Today I want to talk with you about some of the work that we're doing about nutrition during pregnancy, the impacts of that on the health of the infant, and starting to think a little bit more about the impact of maternal health and nutrition on not just survival of the child, but the thriving of that child. And that's really part of the central theme of the presentation I want to give you today. So first, just a brief review about maternal nutrition, some of the consequences of that. Many of you are familiar with these, maternal morbidity and mortality, low birth weight children, a stillborn child. And I want to highlight with you actually this very last one, this developmental compromise in children, and not just the physical developmental compromise, but also the socio-emotional aspects of the child health and the early childhood development, the cognitive development also of the child. So currently, as far as maternal nutrition goes, there are few government programs actually focusing on specific nutritional supplementation. The main one that we're all aware of is actually prenatal iron and folic acid. And this is global policy. Most countries recommend during pregnancy that women receive, in fact, a supplementation with iron and folic acid. So here from Indonesia actually is the typical supplement that was being provided at the time we started some of our research in Indonesia. So our question was, us and others, is this really enough, right? Is this, women are getting this, but they're probably deficient in other things. So what is the case, in fact, for improving supplementation with other micronutrients, so the multiple micronutrient intake? We know that in most cases, dietary intake is below the RDAs. We know deficiencies don't occur in isolation, and that there are synergies between the different micronutrients, so that probably giving them together may have additional benefit beyond iron and folic acid alone. So we decided we would actually evaluate this in a large-scale RCT with the goal of comparing maternal multiple micronutrient supplementation compared to the existing policy of iron and folic acid. We did this trial in Indonesia. I'll show you more details on it in a few slides from now. But we enrolled approximately 32,000 pregnant women, which covered approximately 80% of this population at the trial site in Lombok, Indonesia, and this was done between 2001 and 2004. So I'll show you some of those data and show you some of the recent data actually from a 10-year follow-up of those kids. First, let me just show you what the formulation was that we actually gave to the moms. This was the formulation you can see there. This was designed actually in a group of experts getting together in 1999, I believe, at a meeting for formulating what they thought would be a good multiple micronutrient supplement. Most of these amounts are around an RDA, so it wasn't a deep scientific review, but it was the general consensus of a team of experts of what they felt would be required. And you can see there our iron and folic acid supplement that was provided. This was a double-blind placebo or double-blind control trial, so iron folic acid was the control. And you can see the iron dose and the folic acid dose actually is the same amount as what was in the multiple micronutrient. And that was a special exemption that we were able to get from the FDA of Indonesia, so that we would have greater comparability between the two groups. And you see there the package of these supplements we had manufactured in Indonesia, individually wrapped, and the appearance of the supplement, the size, and the packaging was actually developed through focus groups with the pregnant women themselves. So we designed a product and had it produced exactly for their specifications, and this had a very substantial impact on their compliance. So, as I said, one RDA, and MMN and the IFA had the same dose of iron and folic acid. So this was the trial site. For those of you not familiar with Indonesia, you can see there the Indonesian archipelago. And here we see the island of Lombok, right here circled. For those of you who have been to Indonesia, many of you may have been to Bali here's the island of Bali right next to. That is our trial site. And in 2001, there were 2.7 million persons living on this island. And these were some of the typical health and social issues that you see lots of places. A lot of these sorts of challenges: poverty, clear vitamin A deficiency there at that time. And the mortality rate in children, although the official rate published by the government was around, I think, 47 per 1,000 live births. We actually found through our research it was closer to around 110. So that was a big difference there. And of course, there's a lot of political challenges with finding those sorts of outcomes. So one of our challenges here is, we thought we could always do a regular large-scale RCT, but we realized that we wanted information that was more likely to inform policy of what you could achieve in a programmatic context. So we set this challenge to ourselves really is how to create a new paradigm of doing large-scale randomized controlled trials where you can have rigorous field research, but at the same time, it prioritizes on the needs of the communities and governments in a way that you can take results and interpret them in a way that reflects how a program might succeed, right? So our goal was to carry out this large-scale RCT as a program would be conducted. So essentially the way that we did that, we had to create this sort of hybrid structure between programs and research. And I've just listed there, the summit trial there and typical characteristics of a program or a study. So we really tried to combine these. In the beginning, I thought this would be easier to do a large-scale randomized trial in a programmatic context. I later learned it was actually much more complicated because you don't have the direct control on supplementation that you would like to have. You don't have the direct control on some of the outcome assessment that you would like to have. But you have to engage in a lot of capacity building with government staff and a lot of government relations. So to do this process, at that time, this is more than 10 years ago, right? Of course, you have to develop all these detailed maps and so forth, and working together with the government, we had to interview all these informations, the active trained skilled birth attendants, the traditional birth attendants, health extension assistants. We actually had to enumerate 752,000 women of reproductive age. And I only mention these numbers because I think that it's still undervalued or underappreciated how accessible it is actually to do large-scale randomized control trials or large-scale field studies in general. So at this time, this was without hand phones, cell phones, large computer technology. We did this essentially hiring, enumerating all these people in around 4 months, right? So it's all possible. It's only a question of implementation and management and implementation, which is something I think, in public health we could learn more about. And I'll talk about that in the talk tomorrow. So what did we actually do? So once we went through all this, we actually came up with a cluster randomized trial. The skilled birth attendants there were 262 of them that were randomized to distribute either iron folic acid or the multivitamin. And the woman would receive every 30 days a packet of the supplement. She would take them every day up to 3 months postpartum. And we, of course, had to do a lot of social marketing and promotion. So this just gives you an overview of the enrollment process. The woman would self-report to antenatal care provider there on the island. Again, we were not going out directly recruiting or giving supplements to pregnant women. We had to promote the use of government services. So you see here, if she showed up there, she would be given a urine test. We did provide those. It turned out that having a free pregnancy test was the biggest factor influencing women enrolling in antenatal care. So that was an interesting public health lesson from that. But if she was pregnant and she consented, she would get either the iron folic acid or the MMN supplement. Again, it was a double-blind randomized trial. If she refused, she would receive those government iron folic acid supplements. So I'm just highlighting this because this is some information I'm going to come back to later. We did obtain blood spots from approximately 18,000 of the moms who enrolled, and venous blood specimens from approximately 6,000 randomly selected one. And that's sort of a sampling framework. So those women in this blood subsample would get a blood taken at baseline, and then at one of those other 4 time points, either a month after supplementation, 36 weeks gestational age, a week after delivery, or 12 weeks after delivery. We didn't take any specimens from children because if you're doing a mortality trial, you're tracking that, and you have a mortality rate of around 10%, it's likely that some child you had contact with for taking blood is not going to survive. He's going to pass away the next day, and it'll be thought that that's because you took blood from them. And so that would, of course, compromise the whole study. So we didn't take any blood specimens from kids at that time. So here's what happened in the original trial here. We published this study in 2008, essentially showing that multivitamin supplementation during pregnancy results in an 18% reduction in child mortality out to 3 months after delivery, with certain subgroups having a larger effect, and I'll show you some of that in just a second. But what was interesting is that you can see that the survival curve essentially, of course, most children, if they, in the first 72 hours, right, which would be up to about here, that's when most of the mortality is occurring, which you see there. And these curves are more or less the same, up to really around 5 days after delivery, maybe 4 or 5 days after delivery. And then you see this curve start to progressively separate. So once you get out to around 12 weeks postpartum, you see this around 18% change in survival. We looked at longer-term outcomes. We look at cognitive performance in these kids at 42 months of age. We found improvements in cognition, and I'll show you some of that data in just a second. But first I wanted to highlight on this, in the subgroup analysis, what we found was that the multivitamin, again, compared to iron folic acid, appeared to have a stronger effect in women who had a mid-upper arm circumference below 23.5, so if the woman was a thin woman, and particularly striking is if she was anemic. So really I think highlighting that if a woman's anemic, it's not just about being deficient in iron or folic acid, it's just a marker, right, for being deficient for lots of different things. So shockingly, 38% reduction in child mortality in that group. And interestingly, you see this dose effect as well, that if women took 4 capsules or more, they experienced an impact of it. So again, this suggests that probably the dose maybe wasn't enough. It's likely that there needs to be some adjustment there. It's a bit small here, but this is just looking at the effect on birth weight, that again, the fascinating thing here was that here you have the women who were thin that got the multivitamin, essentially no effect on birth weight at all. However, if she was well nourished, the multivitamin did have an impact on the birth weight, 18% reduction in low birth weight. Mean birth weight improved by 37 grams. Not a big deal really, but interestingly that women that were well nourished are the ones that experienced the improvement in the birth weight. And there were several other studies done with multivitamin supplementation, observed a very similar thing, that if a women were better off nutritionally and they got the multivitamin, they're the ones that had a greater improvement in birth weight for the kids. So interestingly here, we see that, and we also see that even if there's not necessarily an impact on birth weight, there's a survival effect, right? So there appear to be a couple different things going on here. So just to highlight some of the other effects on the mom herself, I think in public health we've really thought about the mother, the importance of the mother as a container for the baby, that she herself as a person is not important. So I wanted to show some of the data also of the impact on the mother herself, that if she received a multivitamin, night blindness rates were reduced. And these are self-reported also, heavy bleeding at the time of delivery and illness around birth. So there appears to be an impact on the moms themselves. But what I wanted to show you was something a little bit unusual. I think this was one of the first studies where we actually decided to look at the effect of the multivitamin on cognitive performance of the women themselves, right? Because there's a lot of issues around how nutrition affects the biological health of children or perhaps the woman during pregnancy. But we were interested also in understanding how it might affect her cognition and issues surrounding her potential ability as a caretaker, both for herself and her children. So we looked at this. We're one of the few groups to actually look at this, this cognitive performance. So we enrolled moms. We randomly selected around 640 of them, some while they were still pregnant, some afterwards, and you can see the distribution there. And what we observed is here. So this is showing you the mean cognitive Z-scores. So we tested several different cognitive capabilities of them, then we created a Z-score of those scales, and this is just showing you what the mean Z-scores were. So you can see there that we had a substantial impact, and I'll show you what these effects actually, what they translate to. So in all moms here, we can see that in fact there's around a 0.1 Z-score change improvement actually, if the women got the multivitamin compared to if they did not get that. And that seemed to be accentuated, again, if the woman was undernourished or if she was anemic in some way. And that 0.1 standard deviation translated for them to approximately the change in Z-score acquired through 1 year of education. So it was pretty substantial effect. I mean, so that's not saying that, how do I say that? That's not about the knowledge you get from the education, but about the change in your cognitive processes, right? The change in your cognitive abilities. And then you see a similar, and actually for these anemic women, see if they're undernourished or if they were anemic here, these sorts of changes actually were equivalent to 3 years of schooling, 3 years change in cognition due to exposure to school. So pretty substantial effects here. Similar thing actually being observed with the reading efficiency. So this was, I showed you this because this was the single, of the several different domains that were examined, the reading efficiently independently was statistically significant. All the cognitive domains assessed had a change in the same way. This one happened to be statistically significant, and of course overall there was a significant effect. So we're seeing this improvement in the cognitive ability of the moms, and how that translates into the health of the children is, I think, something that we are looking at now in the long-term follow-up of these kids. I mentioned the 42-month follow-up that we did on a small group of children, around 400 or so, and here we see just the domains themselves, and again the composite domain Z-score of these different outcomes, essentially showing that those kids whose moms got the multivitamin certainly had improved cognitive performance. Also strongly associated with whether the mother was undernourished or not. And so you can see really substantial effects here ranging from around 0.3 to 0.2 Z-scores, particularly in these three domains here: motor development, language development, and visual attention. So from this, we started getting the idea that, look, there's these substantial effects of the multivitamin on survival, on maternal health, aspects of maternal cognition, possibly behavior, certainly on children also at 42 months of age. So we're interested in exploring this more and understanding that. And one of the things that we were curious about was this particular paradox that I highlighted to you earlier, which is that in the thin moms, the multivitamin reduced the death in the infants by around 25%, but that was without any change in their birth weight, right? So the prevailing paradigm at that time, and still to some extent, is that birth weight is extremely important, one of the key predictors of survival, but that in fact, we were looking here at an intervention that appears to have substantial impact on survival, maybe not as big an impact on birth weight as you might expect. It's maybe not commensurate with the effect on survival. So we started thinking about, what is actually going on here? Is the effect of the multivitamin doing something more hidden, I guess, than the effect on birth weight? And so we started thinking in this way, that can we think about the main effect of maternal nutrition or maybe one of the earliest effects, or more sensitive effect of maternal nutrition being on what we call qualitative development of the child. So what are the factors that make a child a robust entity, able to survive and adapt and to thrive? So these are two frameworks we thought about. One is better homeostatic biology, meaning essentially if the individual is faced with a challenge, how do they manage that, right? So it's about adaptation essentially. How do you handle a challenge in some way? And the other one was, can we assess their anatomical development? So certain anatomical structures are ones that you can scale, like blood vessel branching patterns, airway branching patterns, the filtration process in the kidneys, those anatomical structures. And so these are things that you can actually scale the complexity of. So not have a change in the mass, but a change in the functionality of it, right? So we thought maybe these are the ways that the human body is actually scaling itself, depending on the resources. So during development, it's not just about the size, right? It's about the complexity of that organism from both a regulatory standpoint and an anatomical standpoint. And currently, there's very few indicators related to that. In fact, we don't even assess health based on adaptability, right? We just assess health based on, height, international height and weight. If you're lucky, maybe someone's assessing, your C-reactive protein or something in your blood, but it's a fixed measure, right? You're not looking at how that's changing over time. And so these are things we could probably look at a lot more. So we started to exploring some of these things. So this is just giving you a little idea about these homeostatic feedback systems, sort of a diagram here, that in a healthy system, if it's perturbed in some way, you'll see it reaching a new equilibrium fairly quickly. You have a strong feedback loop and reaches that new equilibrium level. If you have loss of feedback strength, so these circuits are talking to each other effectively, but maybe not as good as they could be, you'll see these sorts of oscillation patterns developing and eventually reaching this new steady state equilibrium. And then even greater oscillations and delay in reaching that new equilibrium if the feedback response is not as good or if it's delayed in some way. So our question became, well, if multivitamins are affecting this, is there a way to actually probe that? And so we started thinking, well, is there precedent for this in any way? And we found some very old data. I don't even think you can legally do this kind of experiment anymore. But these were done back in the 60s, like probably 1965, I think. It showed up in this book published in 1973, where they were taking malnourished kids, rehabilitating them. But when the malnourished kids came in to this research center, they would give them a dose of thyroid-releasing hormone. And they would look at the thyroid stimulating hormone response to that. So you can see it here, that what the response is. So there's a couple things you can see here. One is that the heterogeneity here is very high, again, consistent with these greater oscillations. And then once they're rehabilitated, you see the standard deviation or the variation. So this is synonymous with these oscillations I showed you earlier. This is going down. You also see more of this peak and more of these sort of coming down. And you seem to be some of these hyper responders there. So it was interesting. I haven't seen much other data like this, I think for good reason. But then here's the same kind of thing showing. They did this also to these kids. They gave them these insulin injections and look to see what their glucose response to that was. So you see the same sort of thing, right? So here's the initial kids here. This response is sort of a blunted response and this slower recovery. Once they're rehabilitated, you see this sort of a stronger response and recovery. So their homeostatic control system seems to be better developed. And so we started thinking, okay, there's some precedent for this. Can we see this in our kids? So the one thing that we had on these kids looking at homeostasis was their APGAR score, which is the score of the healthiness of a newborn child from the first minute to the 5th minute, right? Just looking at their physiological appearance, essentially, their biological appearance at that time, assessing these key characteristics here. So we just wanted to know that, does it seem like our kids, if the mother got the multivitamin, are they adapting better? So we looked at this. This is just showing you the APGAR scores themselves because we wanted to know if this data seemed like it was reliable. So you can see the 1 minute and the 5 minute APGAR scores the distributions there. They appear to be more or less normally distributed. They're increasing. So we had confidence that we could look at this data. And so our goal was just to see what's the likelihood of there being an improvement in the response in the APGAR score if the child got a multivitamin or not. So you see that here, if the mother got a multivitamin, and the effect is on the child, of course. So you see here about 1.3 relative risk, significant relative risk of improvement in the APGAR, I think of 1 point or more, if the mother had gotten a multivitamin. We had certainly subdomains here on a smaller n, and the only one that seemed to have some indication toward having a change was actually the heart rate, the maturation of the heart rate. And I'll talk with you more about that, not today, but in the talk tomorrow. So this seemed to suggest that, yes, there's some impact, in fact, of the multivitamin on improving the adaptability of the child. So it's consistent with this hypothesis that without size, you can have very substantial changes in the health of the child. Then we started thinking about energy metabolism, right? Because energy metabolism and how that's regulated is, again, a very important part, because if you're going to have greater homeostatic processes and greater anatomical complexity, the energy necessary to drive that is extremely important. So we had these blood specimens from the mom. So the question was, what could we actually look at in there that would be related to that? So this was one of the ideas we had. Let's look at the mitochondrial activity, and the proxy for that was essentially this, the copy number. So the copy number of the DNA in the mitochondria, with the idea being that this gives us a window on the healthiness of the energy metabolism, in the sense that it's been shown that, in fact, during gestation, intrauterine growth restriction has been associated with an increase in the copy number. So in response to stress, oxidative stress in some way, this copy number appears to be increased, so it's suggesting that energy metabolism is being affected. So this is just a paper that was published several years ago, again showing, so this is the copy number of the mtDNA, showing that in pregnancies that were intrauterine growth restricted, the peripheral blood content in the mother of the mtDNA is increased. And we were looking at this because that's what we had. Ideally, of course, you'd want to look in the kids themselves, right? But the specimens that we had were from the mom. So we were encouraged by this, so we thought, well, let's go ahead and look at this. Is the copy number, what's the relationship between the copy number of the mitochondria and whether the mom got multivitamin supplementation or iron folic acid? So unfortunately, this was colored yellow, but if you can see this, essentially what we saw was that the copy number in the mother, if she got a multivitamin, in fact is correlating with a lower copy number in the mom compared to the iron and folic acid. I didn't put the p-value on here, the analysis. With a log rank test, this was significant. I think it was a p-value something like 0.015 or something. So this is consistent with the previous research that was done, that increase in mitochondrial copy number in the mother is associating with greater intrauterine growth restriction. So here we're seeing the MMN, again, consistent with these effects, likely a healthier child, possibly related to the effects of MMN on energy metabolism in some way. So this is something that we want to look at more in the future. So what about this issue of the anatomical complexity? How could we have looked at that? So we decided that one easy way to do this, since we couldn't do a lot with a blood vessel branching analysis, we could have taken ocular pictures. That was a bit of a challenge in the field to look at the blood vessel branching pattern in the eye. But what we thought we could do this was look at the ridge patterns on the hand. And this has been shown to have some associations with intrauterine growth restriction. This area specifically of the hand forms between the 10th to 24th week of gestation. This is known as this AB region. so we thought, maybe there's a way we can take some images of the hands of these kids relatively easily and analyze those and see, are we seeing any associations with whether they got, the mother got the multivitamin supplement or whether or not the child was growth restricted in some way. So with colleagues from MIT, we actually produced a device, a simple infrared, near-infrared illumination device that we would place on the hand, take a picture of that. So part of the 10-year follow-up study that I'm going to show you in just a second was getting these kids and taking a picture of their hand. And then we would do an analysis of the surface texture of that with the idea of picking up patterns of complexity. So a composite pattern of complexity related to the hand creases, other features on the skin, and slightly below the surface, so including any sort of blood vessel structures there. So this is what the image would look like. We would take this Gabor filter analysis approach, which is an approach somewhat state-of-the-art, but been used for a while, that gives you an idea of the texture of the surface. And this is just giving an example of some of the wavelet, Gabor wavelet analysis applied to this image and what it sort of gives you. So you run a wavelet filter on this, and it gives you this sort of a output. So we had about 40 filters like that in different orientations and different frequencies. And then you end up generating a matrix like this, because what you're after is a single index of complexity of this, all right? So we go ahead and we do that, and we execute what is essentially a principal components analysis on each of these images. And we end up getting, through a machine learning analysis, a association is the variance that we can, is the variance of the prediction that we get, is it better than the overall variance if we randomly select from the population about who got multivitamin or not, or who was low birth weight or not, or if the mother was undernourished or not. So what we actually found was that through this initial machine learning approach, that in fact the mother's nutritional status, first, no association with multivitamin or iron folic acid. This is for the whole hand. So our next approach, of course, would be to look at this specific AB region of the hand, which we haven't actually done yet. But what we did find was association with gestational age at birth and if the mother was a thin mother or not with her height or maternal HB level. So the initial analysis of this suggests that we're not seeing a gross impact of the multivitamin on complexity of the hand. We are seeing, however, associations with maternal nutritional status on the complexity of the hand pattern. So let me take you through the re-enrollment study. So this is where we were. Can complexity therefore be a more sensitive indicator of the effects of nutritional status, or as an indicator of the thrivability? And I've taken you through a few examples of that, some of the things that we have looked at. So the exciting thing we had a chance to do then was go back and find all these kids about 10 years later and look at their growth and their cognitive development. So we just recently completed this, just about a few months ago. And we were able to find about 10 years later, around 72% of the kids, there were actually 27,300 of them who were alive, last known alive at 3 months postpartum. And we had a specific way of sampling them, which we took sampling them for more detailed cognitive assessment. And that was based on what we observed before. So what we were doing was taking a random sample overall of these kids that we found, around 840 kids, and then specifically sampling that same number of kids from those cohorts where the moms were either undernourished or anemic at enrollment. Because remember, that was a group that there seemed to be specific effects in, or larger effects in. And of course, we also chose all those kids born of the moms who we tested their cognition of. And we also re-enrolled as many of the kids that we could find that we had assessed at 42 months. So that came out to be around 3,400 children tested for their cognitive assessment. We were told by most cognitive scientists at that time that we were crazy, that you would never do that detailed cognitive assessment on 3,400 kids. But we did it. And so this is just a list of some of the other things that we were looking at in these kids. this palm complexity I talked about, immunological maturation. I'm not going to show you data on that today, but some of these other things, school performance and their cognitive performance. So I'm going to focus mostly on the cognitive performance, and this is just giving you a look at, again, some of the blood specimens that we took and some of the assessments that we did. I think this third bullet point of the accelerometer-based activity and sleep assessments, those data are still under analysis. I think those will be amongst the most interesting, actually, because we'll get to see 1 week activity of kids, well, between 3 days and 1 week of activity of kids having continuous assessment of their heart rate, their stress levels, skin temperature levels, and activity levels. So we're, again, from the standpoint of this homeostatic self-regulation, I think that'll be extremely interesting. So these are the specific cognitive tests that we did on this subgroup of 3,400. This is a typical type of battery of cognitive tests. We had to train, of course, these assessors, standardize them so their inter-rater agreement was fairly high. And let me see if this is, I think this is this, if I can launch this little video here. I don't know if you'll be able to hear. This is just giving you an idea of how the testing was actually done. I don't know if he's, probably won't be able to hear that. In different stations done in schools of these kids. So we would go to the schools, select these kids that were there, and they would be pulled out to have this test done on them, these series of tests done on them. And again, I think this was the challenge in being able to standardize the different teams, because there ended up being something like 68 assessors who were trained and standardized. And that's a huge part of doing these sorts of things, because there actually is quite a bit of variation in the test performance based on the tester. And of course, in this climate, I mean, people had said you have to do this in a what do they call it, like a quiet, soundproof area, which is not practical in this kind of context. So again, assessing what's the impact of ambient noise and all this is important. So just a quick overview of the analysis framework that we were looking at. Many different things to integrate. The mother had gotten the multivitamin or iron folic acid years before. This is just sort of a conceptual model of that and what we were looking at. We saw this pathway before related to the nurturing and the stimulation. This turned out to be extremely important. The home inventory, we found this before at 42 months of age, the richness of the home environment, how the parents interact with their children, what they have in their household, toys, books, other things, extremely important, probably the biggest predictor of child cognitive performance. And then, of course, all this affects these abilities that we assume is going to have an impact on educational attainment in some way, and of course, human capital down the line. So what did we actually observe? Let me walk you through this a little bit. What we observed was that, in fact, there were long-term effects of the multivitamin. Relatively medium to small effects, I would say. So on some of these executive function tests, so overall in the children, you're looking at about around 0.1, here's 0.09, 0.08, around 0.1 standard deviations on the impact of some of these things, looking at executive function. Again, slightly larger effects in kids born of women who were anemic, who got the multivitamin. Okay, so these are all increases in Z-score amongst kids who got the multivitamin. So you see some of these others, educational attainment, correctly recognized sentences, high performance in being able to read a sentence, explain that sentence, in the motor performance, some subgroup having some effects on that also. So overall, looking at around between 0.1 to 0.3 standard deviations. So in a 10-year-old child, these translated into developmental delays or developmental accelerations of around between 3 and 9 months. So not huge effects, not small effects, but interesting that after 10 years you can see something like this from a multivitamin supplementation given during the pregnancy. But now here's the really interesting part, I think. So there's no impact on some of these things overall, no clear impact on overall school performance. We're looking at this more carefully because we believe that these grades, the most recent grade that we got the records on, the sad news there was that it appears that at least somewhere around a fifth to 1/3 of the grades appear to be fake grades from their teachers. So where the child will score in every assessment 60%. And so so we have to go through and filter those out through a probabilistic process, which ones appear to be real grades, which ones are wrong grades, and then go through and redo that assessment. But now here's the interesting part. We also looked at the relative effects of social and physical factors, other factors on cognitive performance, because we had the opportunity to be able to look at that. So these are just give you a look at some of these selected social factors, their gender, home environment, improved toilet, maternal mental health, maternal education, and so forth. And we look at these physical characteristics as well, maternal height, child stunting, because right now there's a lot of interest in cognition, and a lot of the attention has been focused on these physical characteristics, stunting and cognition and so forth. So we were interested, we had the opportunity to see what's the relative contribution of these. And so let me just show you some of these plots. I just made these simple spider plots where I put these physical determinants, so to speak, down on the bottom part here, and these social determinants up here. And I just indicated what was the z-score of the impact, right? The relative z-score of the impact. In some cases it could be negative, in some cases it could be positive. But if it was, and I've indicated if it's significant or not. So this is sort of giving maybe a quick visual way to see what's the relative contribution on the upper part, which is more the social determinants, versus the physical determinants on shaping the cognitive performance. So here you have educational attainment, the arithmetic score specifically, and you can see that, yes, these physical aspects are important. The stunting of the child, that's significant effect there. Preterm birth, significant effect, but smaller, so substantially smaller than any of the social factors. So these social determinants are very important. And let's look at a couple of other of these because they're interesting. So you see that profile there. So here you see another set of profiles on general intelligence from the WIS test, verbal and the nonverbal there. And so what I find fascinating is, yes, you see the dominance of these social determinants, but depending on the test, the strength of various influencers changes, right? Which is part of, I think, the challenge of doing cognitive assessment. You're not just assessing one thing, and different cognitive functions are influenced in different ways by different social factors. And this is what you're seeing actually emerging from this. Here's the Stroop numbers test, which is an indicator of cognitive control. So big effect of gender. Girls tended to do better. This was interesting here also. Girls tended to do better on these things, a very strong impact there. And so I don't want to go through all of these. I know we're running out of time here, but this is the consistent pattern. What we see is typically, now this one I will show you because this really stands out. The maternal mental health has such a strong impact on the socio-emotional state of the child, right? So you can see that. So this really brings us to this issue of what really do we need to do, right? So nutrition is part of it, physical health is part of it, but we're not going to get thriving populations until we address the social part of it. So we need nutrition integrated with social interventions in some way. And this is, I think, one of the big challenges now. So just the overall conclusions there on the 10-year follow-up. Yes, there are long-term effects, medium to small effects, I would say. These tend to be consistent with what we saw, the impact of MMN on the birth weight and on survival. But the real interesting part is really looking at the social factors and understanding that these seem to have very, very strong long-term effects. So what are we planning on next? Some of this stuff is ongoing, actually, looking at the long-term effects of this maternal and early childhood environment on human capital development, and particularly some of the non-communicable diseases. What's happening there? Are we already seeing children at higher or lower risk for either diabetes, cardiovascular disease? We're interested in what specific nutrients are mediating that effect. Of course, it was a multivitamin, so we have to have ways of teasing that out. So we want to look at low responders versus high responders. Possibly look at those individuals segregated by those who have genetic polymorphisms, where they're high metabolizers or low metabolizers, and be able to look at specific effects, identify specific components of the multivitamin in that way. And I want to take you down actually to this last point, assess interactions of behavior and stress on biomarkers. So we often think of it, the direction being there's sort of a biomarker, biological predictive of the prediction of the person's health. And what we're seeing from some of this, our data and also other data, is that the other relationship exists also, right? So their behavior, person's behavior, their stress levels and so forth are influencing their biology. And so that's something we also want to look at more. And so I just want to acknowledge there's so many people that really put so much into all this work. There's some of the collaborators here at the Summit Institute of Development, Beth Prado, Michael Ullman, and Katie Alcock, really huge contribution to the cognitive work. The work that I showed you there was mostly overseen by Beth Prado. Biological specimen analysis, all done in Indonesia because you're not allowed to send any specimens outside of Indonesia. The anatomical complexity really with my colleagues at MIT, Dan Chamberlain Barsha and Rich Fletcher. So thanks for listening. So, okay. And we have a few moments for questions. Sorry I didn't leave more, but yeah.
Questions? Patrick.
I have a question. So I am really interested in the way developmental biologists are thinking about some of this, and that is that menopause isn't restricted to the ovary. That is, that you run out of quality stem cells or a number of stem cells in an important organ. That nutrition, early nutrition, plays a huge role in establishing these stem cell niches, and then the quality of them. And you touched on both of these in terms of some of the outputs you look at in cognition as well as the mitochondrial DNA and relating to quality. But all of your outcomes are sort of snapshots.
Yes.
And you're seeing a small gap. Has anybody looked to see over time? Because if it's a stem cell issue, that gap should then increase over time.
Yes.
So you look at cognition, at 10 years, then 20 years. But is there any longitudinal data looking at those sorts of exposures in pregnancy over time? That'd be more consistent with the biology.
Yeah, there are a few studies where people have started looking at that. That would be the ideal approach. And I think that what our focus here now is actually to take the kids, basically to focus on those specimens that we have, where we have data from the mother, from the kids at 42 months of age, because we also took blood specimens there, and then the kids currently, and at least seeing what we can look at of a continuum along that. I think what you really need to do to address that issue, which is very important, is to just start doing that, right, with new cohorts, following them up over time, because some of those analyses, you have to do those on fresh specimens, right? And I think that in that context, what we've learned is that it's likely that you're going to need to collect a lot more information during the pregnancy and in early childhood than what I think we have traditionally been doing. But I think that's extremely important to look at that. The one thing that you can do, which we've begun doing also, is looking at some of these regulatory feedback pathways. What we can do is look at what are the markers that should have some sort of correlation, positive or inversely correlated, that and do we see those at the snapshot? Because the way we can look at the snapshot as far as this regulation is concerned is if you can't look at it longitudinally, we can at least look to see if the standard deviations are affected and if we're having internal consistency of the biomarkers. So that's our way of looking at it in this population. Yeah.
Other questions?
Other questions? Yes.
Why would intrauterine growth restriction? Can you explain the direction of that relationship, the effect on mitochondria?
Yeah, that's a very good question. That's a very good question. So when we first started looking into this, I thought for certain, right, the greater copy number would actually be a positive thing, right? Because that greater copy number would indicate that there was more energy metabolism going on. But it turns out that the story with the mitochondrial DNA copy number is more complex because that copy number can be increased due to stress, oxidative stress, other stress in some way. And so it appears that from the data published so far, and I think this needs to be looked at more actually, because those studies published are still relatively small sample sizes, but indicating that mitochondrial copy number during pregnancy in the peripheral blood of the woman actually is going down during the pregnancy. Doesn't appear to go down if the child is suffering from intrauterine growth restriction, likely due to, again, additional stress. It's a sign of stress. Now, there are various hypotheses that there are cells from the baby that are crossing over to the mother's circulation that are influencing this copy number in the maternal blood. When I look at the numbers of this, it doesn't seem really plausible to me, but that's something that needs to be looked at. Incidentally, in this population, you could probably see from that one graph that many of the women here, as they were progressing through pregnancy, because we had those two different, what was shown there is the change from the first time point to the second time point, that in fact in this population, at least half of them are having an increase in the mtDNA copy number. Whereas women that were, when this was looked at in the West, they were all having a reduction in copy number during the pregnancy. So again, suggesting that these women are under stress in some way, and the multivitamin reduced that copy number more. But I think that's something that does need to be looked at a lot more.
Can I ask a quick follow-up
Yeah,
to that?
yeah.
Often when you see increases in copy number,
Yeah.
it's the quality of the DNA is terrible because you start getting these mitochondrial deletions. Was there changes in insertion deletions in the mitochondrial DNA in those cases with the IUD?
Those were, I'm not sure about that. What I can, yeah. What I can tell you, I mean, we certainly did was screened for those specimens that had low degradation and ones that we could do the haplotyping on also. So those were data from those. Now, interestingly enough, and maybe in part to your question, is that the copy numbers we were observing were also lower, on the lower side, I should say, from copy numbers that have been reported. You know, certainly within the range, but on the low side. Yeah. So that's something that should be looked at. Yeah.
Maybe one more question. Yeah.
Very, very, very interesting your presentation. So given the results you have, what will be the recommendations you will do for public policy? Now we should give the moms both iron.
I personally, you know, there's controversy around multivitamin supplementation increasing mortality in children in low-income countries, right? There are a few people that have published studies on that. There was a couple years ago, a large-scale trial in Bangladesh showing that they didn't observe that adverse effect. They didn't observe a lot of positive benefits either, but they didn't observe that adverse effect. I think based on our data, and when I look at the data overall, my opinion is that we should go towards this recommendation of the multivitamin. I'm not sure if that's the right formulation, the one that I showed you, but certainly some move towards giving more than just iron folic acid during pregnancy. And I will say, though, I think that's a small part of the story, actually, right? Because it's clear that protein calorie supplementation can also be extremely important. This whole issue of maternal nutrition, looking at that, also in the context of behavioral factors, you know, lower stress, lower mental stress during pregnancy, lower physical stress during pregnancy. So we've actually, I'll show some of this to you tomorrow, is we've created this program in Indonesia called the Golden Generation Program, which is about nutritional and socio-emotional intervention during pregnancy and early childhood, which is focusing on home-based counseling to have a baby-friendly household, which is also intended to have, you know, just to increase general mental wellness and physical wellness in the household. And I think that's the direction we need to be moving in, actually, is thinking more about, again, interventions that directly and indirectly enhance the thrivability of people. You know, what's exciting to me in today's science is that we're getting more integration on lots of levels, but one of the levels we're getting more integration on now is from the mental health standpoint with the physical health.
And this is very interesting because in many countries there is a pervasive culture of protective factors for pregnant women.
Yes.
I mean, there is a social
That's true,
influence that
Yeah.
everybody says, if you're pregnant, we should feed you better, we should take care of you,
Yeah.
this and that.
Right.
And we are losing that,
We're losing that.
as with
yeah.
the urbanization.
And those practices are there, and those are not always practiced also, because the, the woman also has to stand up for herself in those contexts. So all those things need, attention, both from the nutritional standpoint and, the social standpoint. That's a good point, yeah.
Thank you. So that's a great place to stop. I'd like to thank Dr. Shankar for joining us. And also tomorrow, there's his second talk is at 2:30, also in this room. So please join us. Thanks a lot, Anu.
Okay, good. Thank you.